In this video, you will learn about the testosterone signaling pathway as it relates to the development of the male fetus. We will be discussing the testosterone signal, the androgen receptor, and a detailed explanation of how the pathway works. While there are three possible pathways, we will only be covering the classical signaling pathway in this video.
This video will be focusing on the signaling pathway associated with the hormone testosterone, a ligand that travels through the bloodstream. Testosterone, made up of carbon, hydrogen, and oxygen, is a small and nonpolar ligand which allows for it to diffuse through the cell membrane. As a result, the receptors for testosterone are found inside the cell. These receptor proteins are called androgen receptors, which are usually found in the cytoplasm of the cell. The primary purpose of testosterone is development of the male primary sexual characteristics, specifically in the fetus as it pertains to this video.
How is testosterone released? The hypothalamus releases a signaling molecule called GnRH, which binds to the pituitary gland. The pituitary gland then releases luteinizing hormone, or LH, and the presence of LH signals for the release of testosterone into the bloodstream.
Androgen receptors function by binding to androgenic hormones. After being bound, the receptor is translocated to the nucleus where it bonds to androgenic response elements and regulates gene expression pertaining to sex characteristics and muscle growth.
Now we'll be talking about the androgen receptor. The androgen receptor acts as the binding site and transport molecule for testosterone. The gene for the androgen receptor protein is found on the X chromosome. The androgen receptor is able to carry out its function due to its structure that consists of three domains. These domains are the N-terminal domain, DNA binding domain, and ligand binding domain. The DNA binding domain is the most conservative of the three as it is equipped with two zinc fingers that recognize specific DNA sequences. These fingers regulate binding to the promoter and enhancer regions of genes regulated by the androgen receptor, thus allowing for the activation functions of the N-terminal and ligand terminal to stimulate or repress transcription of the genes. Due to the DNA binding domain being so conservative, it has been shown that the binding of selective AREs allows for specific activation of the androgen receptor. The ligand domain and the DNA binding domain are linked together via hinge reaction. The ligand domain mediates the interaction between androgen and the heat shock and chaperone proteins. In conjunction with the N-terminus, the ligand domain works to stabilize androgen receptor.
Androgen receptors are advantageous to the body as they allow cells to receive hormones and regulate gene expression to produce sex characteristics. Testosterone also plays a key role in muscle growth, which is why some athletes use it as an exogenous enhancer for athletic performance. However, there are some mutations in androgen receptors that make them unable to perform their normal functions. Certain mutations lead to abnormally short versions of the receptor protein or abnormal receptors that aren't able to bind to hormones or DNA. These mutations can have effects on the development of a fetus, such as feminization of a fetus with XY chromosomes.
The signaling pathway. Testosterone can be attached to the cell through three pathways, being classical, non-classical, and non-classical G protein. In this video, we will only be covering the classical pathway. This pathway includes the ligand, testosterone, the androgen receptor, and other molecules like DNA. First, testosterone, a small nonpolar ligand, diffuses through the plasma membrane. It is converted by the 5-alpha reductase enzyme in the cell, and the resulting 5-alpha dehydrotestosterone attaches to the androgen receptor in the cytoplasm of the cell. Next, a conformational change occurs and releases the androgen receptor from the heat shock proteins that bind it into place. Then, the androgen receptor translocates to the nucleus, where it binds to androgenic response elements, or zones of DNA that can change the expression of primary male sexual characteristics. When the activated receptor arrives in the DNA of the prostate cell, it sets into motion a series of transcription events which stimulate genes in the cell to give rise to proliferation and causes the production of a number of proteins of different enzymatic functions. Co-regulators are recruited and regulate gene transcription. In total, the classical testosterone signaling process requires 30 to 40 minutes to cause changes in gene expression. As genes are expressed, they produce a variety of growth factors, such as epidermal growth factor, which diffuses out of the cell through autocrine or paracrine signaling, stimulates further cell proliferation in other cells or the affected cell itself. The binding of testosterone to the target cell causes a regulation of protein synthesis. This is especially key to the development of muscle.
When the concentration of testosterone in the blood becomes too high, the signal must be disconnected. The signaling pathway is terminated via a negative feedback loop when there is a high concentration of testosterone in the blood. A signal is sent to the hypothalamus to suppress the secretion of GnRH, thus sending another message to the anterior pituitary gland, making it less responsive to the GnRH stimuli.
Fetus development in boys. Testosterone is very important in the development of boys, starting in the fetus. The first release of testosterone is induced by the presence of the Y chromosome in the fetus. This causes a change in gene expression that induces growth and the development of the Wolffian ducts, accessory glands, and male external genitalia.
Testosterone in men versus women. When boys go through puberty and grow into men, testosterone is the key ingredient in this developmental process. Testosterone causes growth in the penis and the testicles, deepening of the voice, a growth of facial hair, an increase in muscle size and strength, an increase in bone growth and strength, an increased sex drive, and sperm production. In women, testosterone generally maintains reproductive tissues and does play a role in bone growth and muscle growth. However, they possess a significantly lower amount of testosterone as estrogen is the primary hormone. In both men and women, a higher level of testosterone can lead to an increased level of energy. Testosterone is a key hormone in both men and women, but men typically have more testosterone.
Testosterone is a hormone that is vital to the development of humans. The signaling pathway is simple, yet causes significant development in muscle, bone density, genitals, pitch of the voice, and sperm production. Thank you for watching.